Let's continue our look at central synapses. Recall that neurons in the brain are getting many inputs, and some of these are excitatory and some of these are inhibitory, and the job of each neuron is to integrate all this information and then decide if it should initiate an action potential at the spike initiation zone. To understand how these processes happen, we need to look at excitatory and inhibitory synapses in a bit more detail. Let me begin by showing you a typical excitatory synapse. This synapse will have a nerve terminal. There'll be a postsynaptic cell, and here I've drawn it as a dendritic spine because many excitatory synapses occur on dendritic spines. Not all do. There are neurons in the brain that are not spiny, and so there can be excitatory synapses onto dendrites. There can also be excitatory synapses onto other part of the neuron. I'm also going to add in an astrocyte because in the brain and spinal cord, synapses are intimately associated with these supporting glial cells. The presynaptic cell will have many vesicles that will be filled with a neurotransmitter. Here I've gone ahead and specified the neurotransmitter is glutamate because this is the major excitatory neurotransmitter found in the vertebrate brain. So most, but not all, excitatory synapses are going to use glutamate as a neurotransmitter. The postsynaptic cell will have receptors for glutamate, and there will be reuptake transporters in the presynaptic cell in order to reabsorb that glutamate. In addition, there'll be reuptake transporters in the astrocyte because one of the jobs of these glial cells is to rapidly remove excess neurotransmitter from the synaptic cleft. Now let's zoom in even more to look at a glutaminergic nerve terminal. This is the structure of glutamate. You can see it's a small molecule. It's actually an amino acid. And cells can produce glutamate using metabolic pathways in the mitochondria. Glutamate can also be reabsorbed from the synaptic cleft. Regardless of the source of the glutamate, the first step is going to involve loading it into the vesicles, and this process is going to involve a combination of antiporters that will move glutamate into the interior of the vesicle, paying for it by moving protons out, and the ATPase pump, which is necessary in order to establish that gradient for protons. Release of the vesicle involves calcium influx, V snares and T snares. This happens when an action potential arrives at the nerve terminal. The transmitter will then be released in the cleft, and the membrane components can be recycled so that they can be reloaded with neurotransmitter. Let's go ahead and look at what happens to the transmitter. The transmitter is likely to bind to a receptor, and there are several different types of glutamate receptors that are present in neurons. We can divide these into two broad categories: ionotropic receptors, such as the AMPA and the kainate, and the NMDA receptors. These are ion channels themselves. Here's an example of a simple ligand-gated channel. For glutamate, there's also a special class of ionotropic receptors called the NMDA receptors. These are ligand-gated channels, but they have this additional magnesium block, which here I've illustrated with a red star. We'll come back to that later. The second broad class of glutamate receptors are metabotropic receptors. These are G-protein coupled receptors, also called the 7 transmembrane domain protein family. And there are several different classes of metabotropic glutamate receptors. Let's look at how these work, beginning with the ionotropic receptors. Ionotropic receptors are pore-forming channels. Ionotropic glutamate receptors allow passage primarily of sodium ions, although some other ions can move as well. They open when their ligand, glutamate, binds, depolarizing the cell. So here, in the absence of any neurotransmitter, the channels will be closed. What happens when we add glutamate? Well, under those circumstances, the glutamate can bind to the ligand binding site, open up the channel, and sodium ions are free to move into the cell, depolarizing it. Now let's look at NMDA receptors, which is a variation on this. NMDA receptors are also pore-forming membrane proteins. They'll allow passage of sodium, and also they're fairly permeable to calcium. Ion flow will depend on two factors. The first is that there's a gate that will open when glutamate binds to the channel. Here it is. The second is that there's a magnesium block, and that can be removed if the cell is depolarized. So let's look at how NMDA receptors work. If the channel is closed in the absence of glutamate, then there's no way for sodium to move in. Suppose we add some glutamate. The glutamate can bind to the receptor, opening up the channel. But because of the magnesium block, sodium can still not flow through. We say the channel's open but blocked. In order to remove the block, the cell needs to be depolarized. If the cell gets more positive, then the magnesium block can be kicked out of the pore. And under these circumstances, if glutamate binds to the receptor, the gate can open up, and now sodium and calcium can flow into the cell, depolarizing it. So NMDA receptors are important because they allow calcium to enter the cell where it can modulate the activity of a number of proteins. So if we can recruit this ion channel, we might get a different effect than if we only activated the AMPA or the kainate class of receptors. NMDA receptors are also very important because they can act as a coincidence detector. The channel will remain closed unless the magnesium block has been removed. Let me walk you through this. Let's suppose we've got a dendrite that's getting input from two different presynaptic cells. If presynaptic cell A is active, well then glutamate will be released. It will allow sodium to enter through the AMPA receptors, but unless the cell has previously been depolarized, magnesium will block the NMDA receptors and there'll be no calcium influx. What happens then if we activate presynaptic input B? Well, sodium can enter. As this sodium enters, it can kick the magnesium block out of those NMDA receptors. If we then activate within a short amount of time before the magnesium block has a chance to reform presynaptic A, well then not only will sodium enter through the AMPA receptors, but calcium can enter as well. So cytosolic calcium will increase if synapse A is activated shortly after synapse B, and in this way we can tell that those two inputs are paired, and this has important implications in learning and memory. I want to also talk about the other class of glutamate receptors. So these are the metabotropic family of receptors. Metabotropic receptors are integral membrane proteins of the GPCR family, but they do not form pores. They are not ion channels themselves. Instead, they're going to change configuration when the ligand binds. Metabotropic receptors like this are associated with a G protein. Here it is. The heterotrimeric refers to the fact that it has 3 subunits. In this inactive state, the alpha, gamma, and the beta subunits are all associated with the receptor, and the alpha subunit will be bound to GDP. When the ligand is added, in this case glutamate, what happens is that there's a conformational change. And this, because the protein is associated with the G protein, induces the exchange of GDP and picking up of GTP. Following the binding of the GTP, the alpha and the beta gamma subunits no longer remain associated, and instead the beta and the gamma and the alpha subunits can diffuse away. In the case of glutamate receptors, the alpha subunit can go and bind to one of a number of different effectors. It depends on the particular type of metabotropic glutamate receptor, but all of them basically act to initiate some kind of intercellular cascade that can then lead to activation of something inside the cell. What's important to notice here is that the effects of binding glutamate by these receptors will be slower, and that they also might last longer relative to ionotropic receptors. So to summarize, at glutaminergic synapses, glutamate release is going to depolarize the cell. It might activate NMDA receptors, leading to an increase in calcium, and it might also activate cell signaling cascades and have long-lasting effects. We'll need to reset the synapse and remove excess glutamate. This is accomplished by reuptake transporters, or the EAAT, excitatory amino acid transporter family. Different versions of the reuptake transporter are found in glial and neuronal cells, but both versions operate similarly and with a somewhat ridiculous stoichiometry, in which 3 sodiums and 1 proton and a countertransporter of a potassium ion is necessary in order to reuptake a glutamate molecule. Let me add this to the cartoon here. So we see glutamate, sodium, and protons all entering the astrocyte while 1 potassium exits. It's not by chance that I illustrated the activity of this reuptake transporter in the astrocyte. 90% of glutamate reuptake occurs in the glial cells. It would be nice if there was a way for the astrocyte to return the glutamate to the presynaptic cell so it could be recycled, and there is. The astrocytes convert glutamate into another amino acid, glutamine, and then glutamine can be transferred back to the presynaptic cell where, in the mitochondria, it can be reconverted into glutamate. As the glutamate is removed, the channels will close, the magnesium block will be returned, and now the synapse is ready for the next event. Let's turn and look at a typical inhibitory synapse. Here's my cartoon. I've drawn the axon terminating onto the cell body, so remember inhibitory synapses can occur elsewhere in the neuron as well. I've also added an astrocyte. Here I've indicated that the synaptic vesicles are filled with GABA because GABA is the major neurotransmitter found at inhibitory synapses in the vertebrate brain. There are other types of transmitters that can have an inhibitory effect, but most inhibitory synapses are GABAergic. Of course, there'll be receptors for GABA on the postsynaptic cell, and here already you can see that there'll be more than one type. And there'll be reuptake transporters in both the terminal and in the astrocyte. Let's look at the GABAergic synapse. Well, here's the structure of GABA. It's another small molecule, and it may look similar to glutamate to you. This is real. Cells synthesize GABA at the axon terminal using an enzyme that takes glutamate and converts it into GABA. The GABA is then loaded into vesicles in a process that's going to involve the GABA vesicular transporter, also called the VGAT or the VIAAT for the vesicular inhibitory amino acid transporter. And this process will require the presence of the ATPase pump. Vesicles will dock and fusion occurs when there's an influx of calcium. And I want to stress that although release of GABA will make the postsynaptic cell less likely to fire an action potential, release of GABA itself requires an action potential in the presynaptic cell. The transmitter can be released into the cleft, and then the membrane can be endocytosed and reloaded with neurotransmitter. Well, let me take just a little bit of a moment to talk about the GABAergic reuptake transporters. I said before that they were present in both the terminal and the astrocytes, so the synapse will be reset when excess GABA is removed from the cell by reuptake transporters. These are called the GAT, or in humans they're called the AT family of transporters. And there are different versions that are found in glial and neuronal cells. They have a complicated stoichiometry. They move one GABA molecule, two sodiums, and one chloride into the cell. Now let's turn our attention and look at the fates for GABA on the postsynaptic cell, which will involve GABAergic receptors. Neurons have several different types of GABA receptors, and these come in two flavors, ionotropic. This is the GABA-AC class of receptors, and they are ligand-gated ion channels. And they're also metabotropic G-protein coupled receptors that belong to the GABA-B class. GABA-A receptors are pore-forming channels. There are multiple isoforms. They're all pentamers, meaning they have 5 subunits, and they're composed out of several different types of subunits, meaning there's a lot of variety in the types of GABA receptors. What they have in common is that they all allow passage primarily of chloride ions. Let's look at this. In the absence of GABA, the channel is closed, chloride can't enter the cell. But if we add GABA, then the channel can flip open, and now chloride can enter the cell, making it more negative. The GABA-B class of receptors are the metabotropic receptors. They are integral membrane proteins of the GPCR family. Again, they're not pores. The functional metabotropic GABA receptor is composed out of 2 subunits, so it looks a little bit different than the metabotropic glutamate receptors, but the way that it acts is very similar. It'll change configuration when the ligand binds, and it's associated with a class of heterotrimeric G proteins that can initiate or turn off intracellular signaling cascades. So in the inactive state, no GABA is bound. We have the alpha, beta, and gamma subunits associated with a GDP. Now let's look at what happens when we add GABA. Well, the GABA will come, bind to its binding site. That then induces a conformational change, which in turn will promote the exchange of a GTP for a GDP. Once the G protein is bound to a GTP, it dissociates. There are several potential downstream outcomes. In some cells, there's an enzyme called adenylate cyclase. This enzyme is normally turning on a signaling cascade, and in some cells, activation of the GABA receptor leads to the alpha subunit diffusing over, interacting with the adenylate cyclase to inactivate it, which then in turn turns off this cascade. But perhaps most importantly, at GABAergic synapses, there's a role for the gamma and the beta subunits. In some cells, the gamma and the beta subunits diffuse laterally through the membrane, and they interact with a type of potassium channel, opening it up. This allows potassium to exit the cell, making it more negative. Other times, gamma and the beta subunits diffuse laterally and interact with a calcium channel, closing it. Now this prevents calcium from entering the cell, which again makes it more negative. So the actions of the GABA B class of receptors will be slower than the ionotropic GABA A receptors, but like the GABA A class, activation of this cascade can lead to the cell getting more negative. It'll hyperpolarize. Well, let me close by talking then a little bit about synaptic integration. Recall that neurons have many different inputs. Suppose that we would insert a recording voltage close to the spike initiation zone and then record the voltage. In order for that cell to fire an action potential, it would have to reach threshold, which might be about here. Well, suppose that we go and we can stimulate these individual inputs. What would happen in the cell? Well, if we went and we stimulated this input A, that's far away from the spike initiation zone, we would get a small depolarization, but it wouldn't be sufficient to reach the threshold. If we stimulate B, because it's a little bit closer, we might see that at the recording voltage as a slightly larger depolarization. And if we stimulated C, we might get the biggest response here, but still subthreshold. If we stimulate input D, which is inhibitory, the cell would be hyperpolarized. Well, activation of any one of these inputs isn't enough to lead to an action potential. But if we were to activate two inputs, say A and C together, then we can get a large depolarization. And if we were to activate A, B, and C all together, well then through a process called summation, we could reach threshold and the cell could initiate an action potential. However, if we repeated this and A, B, C, and D were all active at the same time, then activating that inhibitory synapse would prevent the formation of an action potential. This information is complicated, and neurons are complicated. When we think about cells in the central nervous system, we should imagine them with many, many presynaptic inputs. This image here is of some of the dendrites in a hippocampal neuron, and each one of those little green dots is showing us where there would be a dendritic spine. So again, there are a huge number of synapses per neuron. What makes things even more complicated is that in addition to the two neurotransmitters that I've told you about, glutamate and GABA, there are many other neurotransmitters present as well. Consider the results of this experiment in which a team decided to quantify the amount of neurotransmitter receptor protein in different parts of the cortex. So I'll show you the data for this frontal lobe. What we can see is that there are a lot of glutamate receptors, again, several different varieties. There are also a lot of GABA receptors, including both the ionotropic and the metabotropic forms. But in addition to that, there's receptors for acetylcholine, both ionotropic, the nicotinic kind, as well as muscarinic versions, which are metabotropic. There are receptors for norepinephrine, for serotonin, and for dopamine. Individual neurons are bombarded with information that they're adding up and constantly summing in order to decide if they're going to fire an action potential. It's complicated, but this complication is what allows brains to carry out incredibly complex tasks.